So acetyl-CoA comes down and attaches to the oxaloacetate. So then you're going to get a 6-carbon chain.
Okay. Mm-hmm.
And then it goes through the Krebs cycle. You don't need to know every step for this class of the Krebs cycle, but you do need to know the products.
Okay.
Okay? So oxaloacetate is going to stay here. So that means we're going to use this guy up, right?
Mm-hmm.
So what is going to be one of the products then? How did we get rid of carbons before?
CO2?
CO2. So we're going to put in 2 O2 molecules and get to CO2. I want to clarify that from this point forward, I'm only talking about one pyruvate molecule.
Okay.
So if they, if on the test or anything, they say a glucose molecule comes through, goes through it, then all these numbers are doubled.
Okay.
Okay?
That makes sense.
Okay. Do you know any of the other products of the Krebs cycle?
Is there, no, that's for the electron transport chain. Never mind. Nope.
So, you're going to get NADH.
Okay, because we always want to get that.
We always want to get that. For each, we get 3 NAD plus in and get 3 NADH out.
Okay.
1 FADH in and 1 FADH2 out.
Okay. I've never heard of that, but okay.
You gotta know about him. He's important too.
Okay.
Everyone has a place. Okay.
Okay.
Then 1 ADP in and 1 ATP out. So if I said a glucose molecule was to, I guess, what is the products of a glucose molecule all the way from glycolysis to this point?
4 carbon dioxide.
So we've got 4 carbon dioxide, however, there's 2 more right here.
Oh, yeah.
So we've got 6
Oh,
carbon
I gotta
dioxide.
count these guys.
And that's why I'm asking it right now, because
Okay.
a lot of times this transition step is forgotten.
Okay.
So for CO2, how much ATP?
2.
So we got 2 right here. Where else do we get ATP?
Oh, before, up in glycolysis.
Uh-huh.
Okay, so we have
2.
4 ATP altogether.
Yeah, so 4 ATP together.
Okay.
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 NADH?
You had 2 up at the beginning,
Okay.
and now you have 3, 6, so that's 8, right?
And you missed 2.
I missed 2? Oh, at the beginning. Oh, I forgot. 8, 9, 10.
So the transition in
So
the Krebs
10.
cycle and glycolysis.
Oh my gosh, I always
2
forget
more.
those. Okay, so
10.
10. Okay. And then we have 1 or 2 FADH2s.
2 FADH2s.
Okay.
Okay.
That's all I have to know? That cannot be all. Really? That's not it. Really?
I can't think of anything else that you need to know off of that.
Okay.
Yeah, that's it.
All right then.
There is, so one thing that's good to know is that when when we can't use sugars or we're out of sugars or whatnot and we're breaking down fats, there's a triglyceride. So you've got your glycerol and your 3 fatty acids.
Mm-hmm.
Fatty acids are made of carbon chains.
Right.
Okay. What happens is when we use energy, first off the glycerol breaks off.
Right.
And it comes into glycolysis right
Which
when we
gives us
have
energy.
that structure.
Okay.
So it comes in at
The-
3 glycer.
So it like skips a
glycerol
couple
nitrate
steps?
phosphate, something like that.
So it skips a couple steps?
Well, because it's not glucose.
Right,
It has a different structure. So it comes in at that point because that's the structure it is.
okay.
And these guys are cut every 2 carbons. And so
Okay.
they're going to come in
And the acetyl-CoA,
right here, and
Oh.
CoA will come attached to it.
okay. So that one is that's the fatty acids.
So glycerol, 3 fatty acids.
Okay.
Fatty acids, so this process is called beta oxidation.
Okay.
And then this part where the glycerol comes in,
Yes.
it's way up here when it comes in. That is called gluconeogenesis.
And did you say this is when you don't have sugar?
Either don't have sugar, can't use it, like if you were diabetic,
Okay.
and depending on which type of diabetic you were, but if you can't take in sugar into your cells,
Mhm.
like say the insulin's not working or whatnot,
Right.
then you can't use the glucose. So you're going to use different forms of energy, either amino acids, proteins, or fats.